what is the importance of immunity, we will be first knowing few of the definitions which are important in this, right? It will be preferable that you write only the words, ठीक है, and समझ लो. First is immune system. We all are aware of this term. And what is it? It is composed of the different cells, the tissues, organs, right? Molecules, all the cytokines, interleukins, complement system, all those, they constitute the immune system. Then immunology, as already explained, is the study of structure and function of the immune system. What is immunity was well explained by your colleague. It is the resistance of the host to pathogens and their toxic effects, right? And what is immune response? We may be coming frequently to these words, and therefore I am explaining the meaning of all that, right? So collective and coordinated response to the introduction of foreign substances in an individual, mediated by the cells molecules of the immune system. So this is the immune response. So what is the function of this immune system? Defense against the microbes mainly, right? Defense against the growth of tumor cells, है ना? नहीं तो body तो continuously renewal of the cells is occurring inside the body. Most fast dividing cells are present at, anyone? GI epithelium, as well as bone marrow, but it is GI epithelium, right? If you do not clean your tongue daily, there will be a white coating, which are the dead cells. So that coating is occurring in the entire GI, right? Then it is also important for maintenance of homeostasis. Lot number of cells are being destructed continuously in our body for cell renewal, and this immune system is again playing an important role for this maintenance of homeostasis. Anyone, what is homeostasis? Maintenance of a constant internal environment. Okay, so we all are aware of what is composing the immune system. These are the organs. Any organ which you are having any query for, ki how this is acting as a organ related to immune system. Tonsils and adenoids over here, right? Then thymus is there. Then we have different lymph nodes. Then spleen, we all know. Peyer's patches are present in the GI, right? In the intestine. Then we have the appendix, lymphatic vessels, and bone marrow. Okay? Then we have discussed all these cells which are playing a great role in maintenance of immunity, right? Then we have certain molecules, of which we have heard about certain cytokines and interleukins, which are also playing a role in development of this hemopoietic cells, and few are important for this immune system. Okay? Then we have the antibodies, the complement system, and the interferons, right? So as rightly told by you, we'll be discussing this first. One is innate immunity and the other one is the acquired or the adaptive immunity. What is the difference between the two? Anyone? Very good. Very good. फर्स्ट लाइन ऑफ डिफेंस. सो इसके एग्जांपल्स बता सकते हैं आप इनेट इम्यूनिटी के? फॉर एग्जांपल सबसे पहले सेल्स पे ही चले गए. बैरियर्स, देन सिक्रीशंस, है ना? even the tears, saliva, the different enzymes, HCl in the gastric, secreted by the gastric mucosa, they all are the part of the innate immunity, which is present since birth, right? Acquired is anything, or adaptive is anything which the baby or the person acquires with experience or with the invasion of the pathogens. Why the COVID-19 was so deleterious? Because we did not had an exposure to that particular viral infection before. वो पूरे उसके लिए, कम्युनिटी के लिए ही नया था. एक चीज होती है हर्ड इम्यूनिटी. What is, anyone, have you heard about this herd immunity? Okay, we'll be coming to that part also. So this is the first line of immune response, and it relies on the mechanism that exists before infection, so by birth. Then acquired is a second line of response and relies on the mechanism that adapt after infection and are mainly handled by the T and the B lymphocytes. So this innate immunity can also, किसी किसी को, वैसे तो जनरल उसमें बोलते हैं कि इनकी इम्यूनिटी बहुत लो है, but it generally indicates their genetic makeup. Few allergies are inherited from the parents, right? So this is based on the genetic makeup of that particular person and relies on the already formed components. Rapid response within minutes to hours, and it is non-specific as told by him and has no memory. ठीक है? Which is an important part which is present in the acquired immunity and does not lead to clonal expansion. ठीक है? इसका क्या मतलब होता है क्लोनल एक्सपेंशन? अपने जैसी और सेल्स बनाना या अपने जैसे और मॉलिक्यूल्स बनाना, राइट? क्लोन्स. ठीक है? So we have the mechanical barriers, then we have the humoral mechanisms, and then we have the cellular defense mechanism. Every person since, or every child since birth is having all the types of cells, right? So they all are composing of this innate immunity. Now this is the complement system. I just briefed a little yesterday. What is this complement system? They are plasma proteins. Again, they are present in the blood itself, but in an inactivated form. And whenever a pathogen enters in the body, depending upon its antigenicity, any of the three pathways may initiate its activation, leading to cell lysis, right? So we have the classical pathway, which is activated by the antigen antibody complex, right? Then we have the mannose binding lecithin, okay, and this is activated by binding of this lectin to the mannose group present on certain bacterial walls, right? And then we have the alternative pathway, which is directly activated by certain viruses or other pathogens, right? So what is happening is, these are the three pathways, okay, and this classical pathway is acting by conversion of inactivated complement factor 1 in presence of factor 4 and 2 to converting of activation of factor C. Each and every these pathway are ultimately causing what? Activation of C3. ठीक है? Activation of C3, as in the coagulation pathway, the intrinsic and extrinsic pathway were ultimately causing what? They were causing activation of prothrombin to thrombin for further coagulation. So same is happening over here. Now this activated factor C3, this forms C3b, and this causes activation of C5, complement factor 5. This complex of C5, 6, 7, 8 and 9, ठीक है? This complex of activated complement factors is responsible for lysis of the cell. ठीक है? क्या mechanism होता है वो हम बताएंगे, but this complex is causing what? It is causing the lysis of the cell. Now यहाँ पे C3b factor था, इसका जो C3a है, right, C4a and C5a, they are acting as a chemotactic factors. They are responsible for activation, attraction of the lymphocytes towards and neutrophils towards the site of infection. So they are acting as a chemotactic factor. Additionally, they are leading to reactions, that is anaphylaxis. It is a type 1 hypersensitivity reaction, and it is you will be learning later, but it is anaphylaxis. एकदम से बहुत सारा immune response हो जाता है, the person may land up into shock. ठीक है? So these are responsible primarily for chemotaxis, but may lead to this also. And then we have the C3b, which is also responsible for opsonization. In the last lecture, we have learned what is opsonization. So it is making the different pathogens or dead and decaying material palatable by the phagocytic cells. Which are the phagocytic cells we learned? Macrophages, neutrophils, monocyte macrophages we say, right? Okay, so this is the complement pathway. Then we have the adaptive immunity. This complement pathway was for innate response. ठीक है ना, ये already present है. जैसे coagulation pathway में clotting factors already present हैं inactivated form में, वैसे ही ये भी already present है, right? जो different types of cytokines हैं, interleukins हैं, they will be synthesized later because of this, some of them because of this adaptive immunity or acquired immunity. So this is based upon the resistance acquired during the life and relies on the genetic events and cellular. कोई भी प्रोटीन सिंथेसाइज होगा उसका जेनेटिक मेकअप तो हमारी बॉडी में होना चाहिए ना, देन ओनली द डीएनए विल convert into RNA and RNA will lead to the formation of the proteins, right? So that is meant by this line. Then responds more slowly, क्योंकि उसको synthesize करना पड़ेगा, right? उसको synthesize करना पड़ेगा. It is not already present. Slowly we acquired resistance or immunity against the COVID-19, right? But earlier it was devastating, right? So that is the thing. It is specific, जिसके लिए develop होगा most of the time till it gets pathologically converted into autoimmunity. ठीक है? So it is mostly specific, जिसके लिए develop होगा. अगर bacterial infection के लिए है तो bacteria को ही kill करेगा, viral infection के लिए है तो virus को ही kill करेगा, tumor cells के लिए है तो it will be detrimental for those cells only, right? And repeated exposure leads to faster and stronger response because they preserve memory cells. ठीक है? They have memory cells, but innate immunity में ऐसा memory cells type का कुछ नहीं होता है, right? Then it leads to clonal expansion. इसलिए secondary response बड़ा होता है, right? So we have learned that all the cells are synthesized inside the bone marrow. Now these can be lymphoid progenitor cells, which will be forming the lymphocytes. Lymphocytes we divided into two types. It can be divided into two types: small and large. Morphologically and functionally, they can be divided into B cells and T cells. Now this nomenclature of B cells and T cells is dependent upon where they are maturing, right? If they are maturing inside the bone marrow itself, okay, they are known as B lymphocyte. Though the term B lymphocytes, B was given for bursa of fabricus that is present in birds, right? But the one which are getting matured in the thymus are getting the nomenclature of T lymphocytes. Okay? Now these two types, when they are exposed to antigen, ठीक है, ये दोनों circulation में है, B lymphocytes and T lymphocytes. When they are getting the exposure of antigen, what is happening? They are becoming activated T cells, and they are forming different types of T cells. It can be cytotoxic T cells, it can be helper T cells, right? ठीक है? And NK cells are already present. ठीक है? They are the part of the innate immunity. Then we have the B lymphocytes. B lymphocytes, we learned that they transform into plasma cells. And if you see the pictorial depiction, they are larger in size as compared to the lymphocytes. ठीक है? And these plasma cells are responsible for formation of antibodies. The antibodies which are formed will depend upon the type of infection, and जिस टाइप का पैथोजन होगा उसी के अगेंस्ट ये एंटीबॉडी फॉर्म होंगी. ठीक है? तो जो जिसकी वजह से ये एक्टिवेट हुआ है, द एंटीजन विच विल बी एक्टिवेटिंग इट, only to that antigen these antibodies will be acting against. Am I clear? Okay. ठीक है, this all I have already covered, right? So this is I'll be coming to that. Now this is the differentiation between the B cells and T cells. One is occurring in the thymus, the other one is getting matured in the bone marrow. So what is happening in the presence of different growth factors, these interleukins, right? They may be converted into NK cells or into the B cells precursors, right? Now this B cell precursors, they will lead to formation of naive B cells. Naive B cells matlab they are not having any antigenic exposure. Naive means naya, है ना? Like you people are naive to this environment. ठीक है? And depending upon the type of antigenic exposure, these B cells will be developing into specific plasma cells releasing a specific type of antibodies. एक type of plasma cell will be producing only one type of antibodies. ऐसा नहीं होगा कि वो IgG भी produce कर दे, GM भी कर दे. No, right? And then particular जो अभी will be coming to that part, the variable factors होंगे antigen को detect करने के लिए, those will also be same so that they detect the same pathogen as antigenic, right? So this is to be understood, right? Then we have the T cells, and in T cells again, once it is activated, it leads to formation of T helper cells and cytotoxic T cells, which are further depending upon the type of this MHC molecules and एंटीजन प्रेजेंटिंग सेल्स कौन से एंटीजन इनसे एक्ट कर रहे हैं उनके अगेंस्ट ये इम्यूनिटी प्रोवाइड करेंगे, राइट? ओके, सो दिस वी हैव ऑलरेडी एक्सप्लेन. वन थिंग इज, बाकी डिटेल्स में आप ना जाएं अगर, this virus and bacteria or the tumor cells are being presented through the APC. What is APC? Antigen presenting cells. What are the different antigen presenting cells? Macrophages, B cells, dendritic cells, glial cells, Kupffer cells, right? Dust cells. ठीक है? So these are all the antigen presenting cells. ठीक? What these antigen presenting cells are doing? They are presenting this antigen to naive T cells. Naive T cells जो अभी activated नहीं हुए हैं, right? Now we are seeing that they are dividing into two, Th2 and Th1. One is cytotoxic, ठीक है, and one is helper cells, ठीक है. And these helper cells will further activate the B cells. The B cells can also act as antigen presenting cells. B cells भी antigen presenting cells का role play करते हैं. But these helper cells, they help the other these cells for proliferation and clonal expansion. While the cytotoxic T cells, as the name suggests, they are cytotoxic. They kill the inflamed or the infected cells, right? Okay. इसमें एक चीज बतानी थी, it just slipped out, right? ठीक है. Okay. Okay, now this adaptive immunity कितने टाइप्स की हो सकती है? Active as well as passive, as well as the natural immunity can also be active immunity and passive immunity. ठीक है. Natural immunity में क्या हो जाएगा? Natural immunity में जो आपके जितने भी secretions हैं, these are naturally present, right? Passive immunity उनमें कैसे आ रही, but these are not being formed inside the body of that particular baby or organism through breast milk, या फिर जो हम ये करते हैं प्लेसेंटा के थ्रू जो क्रॉस कर गई, राइट? इसलिए अगर हमने मतलब कभी आपने ऑब्जर्व किया होगा, द बेबीज आर डू नॉट गेट, वैसे तो हमें बेबीज को थोड़ा प्रोटेक्ट करके ही रखना पड़ता है, न्यूबॉर्न बेबीज को, बट दे डू नॉट गेट certain type of infection till they are 2 to 3 months of age because of these antibodies already present in them which has crossed through the placenta from the mother, right? Then artificial immunity कैसे आ सकता है? through the vaccination, है ना? And this passive immunity is because of presence of these cells, immune cells, right? So these, coming to the adaptive immunity. Adaptive immunity में दो चीजें हैं, one is cell mediated and the other one is humoral. Cell mediated is mainly because of the T lymphocytes, while humoral is because of the B lymphocytes. Humoral का मतलब होता है blood में. Cytotoxic T cells क्या कर रही हैं? They are killing inside the tissues, right? So these eliminate intracellular microbes that survive within the phagocytes or other infected cells. तो उसमें जाके ये kill कर दे रहे हैं. So they are known as cytotoxic or cellular cell mediated immune response. Humoral immune response is because of the B lymphocytes and is because of the antibodies which are generated by the plasma cells, and these eliminate the extracellular microbes. Cytotoxic में क्या हुआ? जो ऑलरेडी फैगोसाइट्स में है, राइट, ये उनको जाके किल कर रहे हैं, इसलिए हमने इनको साइटोटॉक्सिक या सेल्यूलर इम्यून रिस्पॉन्स बोला. ठीक है. इसमें क्या हो रहा है? ह्यूमोरल में क्या हो रहा है? ये एंटीबॉडीज जो सर्कुलेशन में भी पैथोजेंस हैं, एक्स्ट्रा सेल्यूलर हैं, उनको जाके किल कर रहा है. सो दे आर एक्टिंग इन अ डिफरेंट मैनर. ठीक है. ओके. So these plasma cells lead to formation of 5 different types of antibodies, right? While these T lymphocytes, they form 3 types of T cells. Helper T cells, which is known as CD4 T cells. Cytotoxic T cells, which is CD8. Now what is this CD4 or CD8? Cluster differentiation इसका full form होता है, right? And these are the specific recognition patterns over the cell. ठीक है. And this is responsible for the nature for which they are acting against. ठीक है. So these are mainly tagged with CD8 and these are CD4 T cells. ठीक है. And then we have the memory cells as well. Now, actually this I'll be coming it to later, right? Because मुझे काफी सारी चीजें explain करनी पड़ेगी. What is this? Structure of an antibody. What are the different chains? This is the heavy chain, right? This is the light chain. In one antibody, right? These two chains will be same. Heavy chains के ही basis पे हम इसको name करते हैं G, M, A and D and E. It is gamma, epsilon, alpha. Then M is pentameric structure, and which is D is delta, right? And L भी आपका एक एंटीबॉडी में दोनों चेन सेम रहेंगी, right? Now these two chains are bound by disulfide bonds, ठीक है? And then these two chains also have a variable region. These vary as per the requirement, ठीक है ना? And these are not this variable portion is responsible for binding with the antigens, right? While this portion is responsible, this is the common portion, right? So this is now how it is happening. This, if we see this diagram, what is happening, the this is the B cells, right? We have the antigens, ठीक है? And we have these antigens will be डिटेक्टेड. पहला तो चीज है ना कि वो डिटेक्ट होने चाहिए कि ये एंटीजन है. ये डिटेक्ट कैसे होंगे कि ये एंटीजन है? नॉट जस्ट सेल्फ बॉडी सेल, डेफिनेट पैटर्न्स, पैटर्न रिकॉग्निशन, ठीक है? this is the quality of our immune system which recognizes the patterns of these antigens or molecules. अब these antigens क्या, what all can act as an antigen? It mainly are the proteins, but it can be glycoproteins, it can be lipoproteins, it can be polysaccharides, but most of the times they are proteins, right? So they can detect any of the patterns in the viral infection, in the bacterial infection, any of the pathogens. So these are perceived by the B cells. Then what is happening? These B cells are internalizing it, ठीक है? And there is presence of, what is MHC? Major histocompatibility complex. Okay, this is the gene for that major histocompatibility complex. हम इसको HLA भी बोलते हैं, ह्यूमन ल्यूकोसाइट एंटीजन, ठीक है? हम इफ वी टॉक अबाउट ऑर्गन डोनेशन तो हम कहते हैं ना HLA टाइपिंग करानी है. आपने ये टर्म कहीं पे सुना होगा, राइट? दैट HLA टाइपिंग इज इफ द सेम टाइप ऑफ HLA human leukocyte antigen is present, then the organ donation will be successful, right? So this major histocompatibility complex, इसके लिए जीन प्रेजेंट होते हैं क्रोमोसोम 6, short arm of chromosome 6, and we have the 3 types of MHC gene, MHC1, MHC2, and in between we have the MHC3, ठीक है? Now what this MHC class 1, they are responsible for formation of different types of antibodies, right? These and these MHC1, these are present on all the types of cells, all the nucleated cells except RBCs, उसमें nucleus नहीं होता है, right? So these MHC class 1 is present in all the cells, ठीक है? इसलिए हम, और इसको सबसे पहले ल्यूकोसाइट में डिटेक्ट किया गया था, इसलिए इसको HLA टाइपिंग भी बोलते हैं, राइट? देन वी हैव द MHC type 2 and these are present expressed on the antigen presenting cells like B cells, ठीक है? And the dendritic and the macrophages. Now coming back to, and MHC3 are different from these two, and they are mainly responsible for formation of the complement proteins, the cytokines, the interleukins, heat shock proteins, and all that. So coming back to that particular diagram, वहां पे मुझे इसलिए जाना पड़ा because I wanted to let you understand about the MHC2, ठीक है? So B cells being the antigen presenting cells, the type of MHC present over this is MHC class 2, right? Now this is recognizing it as this MHC is letting the B cell recognize that this particular pattern is not of self and these are antigens, and there the processing of antigens occur. अब एंटीजन की प्रोसेसिंग में क्या होगा? सबसे पहले डिटेक्शन, देन इंटरनलाइजेशन, इंटरनलाइजेशन के बाद अब हम पूरा का पूरा वायरस को तो हम एंटीजन नहीं रिकॉग्नाइज कर रहे हैं ना, वी आर रिकॉग्नाइजिंग सर्टेन प्रोटीन पैटर्न्स और सर्टेन सेल्यूलर पैटर्न्स ओनली एज एंटीजन्स, ठीक है, तो उसका डिग्रेडेशन होगा इन दी वैक्यूल्स, ठीक, and those that degraded product is then further along with the this MHC molecule which is present inside the or synthesized inside the B cells will be presented as a molecule over the B cells. देखो, I may be little bit fast but it's very simple. सबसे पहले बी सेल क्या है? दे आर एंटीजन प्रेजेंटिंग सेल्स. If they are antigen presenting cells, the type of MHC which are present over these antigen presenting cell is MHC class 2. ठीक है? Now the antigen has entered, the pathogen has entered the body. These will be recognized by the B cells. ठीक, रिकॉग्नाइज्ड हो गए, लेकिन इनका नाम क्या है? एंटीजन प्रोसेसिंग सेल्स. And immune response will only be generated by the T cells when this antigen which has invaded our body is presented in a proper form to these cells. ठीक है, एंटीबॉडीज तो ह्यूमोरल, उन्होंने दनादन गोलियां चला दी जैसे ही इन्वेडर्स एंटर किए, बट अब वो एक बार हमारी कंट्री में घुस गए, राइट, हमारी बॉडी में घुस गए, तो हाउ वी विल बी आइडेंटिफाइंग? दे हैव इंटरमिंगल्ड विथ आवर ओन सिटीजन्स, हमारी बॉडी की सेल्स के साथ वो इंटरमिंगल्ड हो गए, राइट, तो हम कैसे उसको रिकॉग्नाइज करेंगे? सो वी विल बी रिकॉग्नाइजिंग बाय दिस प्रोसेसिंग ऑफ दैट पर्टिकुलर एंटीजन by the antigen presenting cells. Over here it is being explained with the help of the B cells. It can be any macrophages, alveolar cells, any cells. ठीक है, अब क्या किया इन्होंने इंटरनलाइज कर लिया बी सेल ने, उसको डिस्ट्रैक्ट कर दिया, उसके छोटे छोटे पार्ट्स बना दिए, ठीक है, लिसिस कर दी. उसमें से कोई पार्ट एंटीजेनिक था जिसने उनको what we were talking about pattern recognition, protein pattern recognition, which was recognized as एंटीजेनिक, ठीक है, जिससे उनको पता लगा कि नहीं ये तो सेम नहीं है, ठीक है, they were not having the same आधार कार्ड आइडेंटिटी which has been given to all the citizens, ठीक है, तो उन्होंने उसको पहचान लिया, right. Now this along with the MHC2 which is present over the cell, इन दोनों का complex will be presented to the T cells. On the T cells, we have T cells receptors, TCRs. We also call it as TLRs, tall-like receptors, and they were primarily detected in Drosophila, but later they were found in our own cells, right? Now these tall-like receptors, these T cells are also having specific receptors for particular type of antigen which is being presented, processed antigen which is being presented by the antigen presenting cells, and then this leads to activation of those T cells, leading to formation of T helper cells, cytotoxic T cells, and the memory cells. Is this clear? This is the entire mechanism of immune system. Anyone who will explain me this? That boy. थोड़ा तेज बोलो, I am not. सुनाई पड़ रहा है, तेज बोलो. Good. प्रोसेसिंग में क्या क्या चीजें आ गया, उसको लाइसेस करके छोटे छोटे टुकड़ों में बना दिया, राइट, एंड देन सम ऑफ द प्रोटीन्स और पॉलीसेक्राइड्स वर डिटेक्टेड एज फॉरेन एलिमेंट्स, है ना, एंड देन व्हाट, how they will be presented to T cells. एक important step galloped. How they are being presented, ये पिंक कलर का बड़ा important structure है MHC, जिसके लिए I went back. It will be presented with the help of these MHC molecules. ठीक है? Then only that will be recognized by the T cells. Otherwise वो recognize नहीं करेगा. Right? So it will be in combination of these MHC molecules. Right? Okay. Except the NK cells. उनको एक्सपोजर नहीं हुआ है. They will only recognize once it is being presented in a proper form to them. जज के पास आप कब लेके जाते हो? जब जज कब कहता है कि इनको हैंग कर दिया जाए या इनको बरी कर दिया जाए या इनको मतलब कारावास, आजीवन कारावास दे दिया जाए. So those are the judges and these B cells are the policemen. उनको with documentation present करना है कि देखो इनका ये पैटर्न रिकॉग्नाइज नहीं हो रहा है. दे आर नॉट द सेल्फ बॉडी के सेल्फ प्रोटीन पैटर्न नहीं है. दीज आर डिफरेंट. ठीक है? दीज अपीयर टू अस एज इन्वेडर्स, राइट? एंड देन दीज साइटोटॉक्सिक टी सेल्स एंड हेल्पर टी सेल्स विल कम इन टू प्ले. राइट? आर यू ऑल अवेयर ऑफ दिस एंटीबॉडीज? ठीक है? तो आई एम इसको एक बार प्लीज पढ़ लेना. एंड I am leaving this part, but this is important. अगर immunity पे आपको question आता है, then you have to mention all these. And the which one of the IgG and IgM which crosses the placenta? And then which is responsible for primary and secondary? IgM is and IgG is secondary. ठीक है? इसको ऐसे भी याद रख सकते हो. So I'm not going in detail of any of these, right? This IgM and you have the IgG is also formed in the primary response, but the amount is less. But whenever there is secondary exposure or invasion of that particular pathogen, then this secondary response is of higher in nature and it is of mainly IgG type, right? Now, substances that can initiate an immune response is antigen. We all know that, and most of them are proteins. But the specificity of an antigen is due to the specific areas of molecules which are known as the determinant sites, and these are known as epitopes. ठीक है? इन्हीं को recognize करना होता है body cells को कि they are antigenic, right? So these are the specific piece of antigen to which an antibody binds. This I have explained. MHC gene, right? It is having 3 parts, class 1 and 2, and third is in between the 2 and 1 and 2, right? These class 1 are present on all the nucleated cells except the B cells. Then class 2 is expressed on the antigen presenting cells, and class 3 are responsible for formation of all these. Now what is the difference between this MHC protein? आपने ये देखा था ना कि वो MHC2 was bound or presented on the surface of the B cells. Earlier it was present inside the B cells, and after the processing of the antigen, it got fused with the antigen and was presented on the cells. Now this MHC class 2 is made up of 2 alpha units and 2 beta units, right? While MHC class 1 is composed of 3 alpha units and 1 beta chain. ठीक है? ये इनका structural difference है. So if you see this is MHC class 1. It is having 3 alpha chains and 1 beta 2, while this is MHC class 2, which is having 2 alpha chains and 2 beta chains. Why this cleft? This cleft is for binding of the particular antigen. ठीक है? Over here also we were having this peptide binding cleft, right? So this is just the depiction of this particular MHC1 and 2, how they are inserting to this cleft. What I want to let you know is what are that diagram, right? What is immune synapse? जिसका हाथ जिसका थोड़ा नीचे है, प्रांजल का, प्रांजल बताएगा. What are immune cells? Immune synapse, sorry. साइनेप्स का मतलब दो चीजें जहां पे कनेक्ट हो रही हैं, राइट, कनेक्शन. So this is the immune synapse. What is happening? Now we have learned about the antigens and how they are processed, right? Now this processed antigen along with the MHC molecule is presented to the T cells. ठीक, these T cells will, these do not only, यहाँ पे एक चीज missing है, that is the adhesion molecules. CAM के बारे में पढ़ा है आपने? What is CAM? Cell adhesion molecules. So this is not the only connection between the antigen presenting cells and the T cells. ठीक है, ये MHC molecule along with the antigens which is presented to the T cell is not the only connection. We have the cell adhesion molecules as well. अगर ये cell adhesion molecules नहीं होंगे, then activation of T cells won't happen. That is important, right? That is important, right? This is the thing. So we have this is antigen presenting cells. It is presenting the antigen with the help of the MHC molecule, and along with that we have the cell adhesion molecules or proteins, CAMs, which will cause linking up of the plasma cells or the antigen presenting cells with the T cells, leading to their activation. So this is composing the immune synapse. Is that clear? Okay. And we have the T cell receptors on the T cells, which is combining with, which are specific and will combine to specific proteins presented through the MHC proteins. ठीक है, ये TLRs भी जो होते हैं, these are also specific. ठीक है, इसीलिए ये पैटर्न को रिकॉग्नाइज करते हैं. ठीक है, so this is just to show you, this diagram is just to show you how this antigen fragment is attaching to the MHC molecule, right? ये कौन सा MHC1 है कि 2 है? 1 एक टांग पे खड़ा है and 2 is having 2 foots. And then this helper cells mainly combine with the MHC class 2. ठीक है, while the cytotoxic T cells, they are combining with MHC class 1. ठीक है, अगर आप दोनों को मल्टीप्लाई करो, 2 into 4 is 8 and 1 into 8 is 8. ठीक है, so this CD4 cluster differentiation 4, which is identifying the helper T cells, है ना, they are mostly acting or are becoming activated by all the antigens which are being presented to them through MHC class 2 molecules. जितने भी एंटीजन प्रोसेसिंग सेल्स होंगी, एंटीजन प्रेजेंटिंग सेल्स होंगी जिसमें कि MHC class 2 होगा, so all will be activating this, right? Then we have the MHC class 1. ये कहाँ पे प्रेजेंट थी MHC class 1? On all the nucleated cells except the red cells. So these will primarily be activating the cytotoxic T cells, right? And this along with the this CAM, they will be forming the immune immune synapse. Is it clear? Anything which is not understood till this? So कल का मैंने कुछ एक काम दिया था आपको. How many have you have made that chart? Very good. Those who haven't made, it's their responsibility. Now one more important thing is you will be learning about all the types of antibodies and will write in short about them in your notebooks so that it is known to you, right? Okay, I don't think anyone who can explain me this now. इसका नाम भूल गई मैं. What's her name? That blue bottle. Your name I just forgot. Gauranshi. Can you explain this diagram? These are the antigen presenting cells. Over here it is the macrophage. So which class of MHC? MHC class 2. All the antigen presenting cells are having MHC class 2. Next, what it is happening? This is processed. Now what is happening? Which T cells? दोनों का multiplication कितना हो रहा है? 8. So if they are having the MHC class 2, they are presenting to CD4. ठीक है ना? Then what? CD4 मतलब कि T helper cells. Then what? What is happening over here? It is activated T cells. These T cells will only be activated if they are having what? मैंने बताया था ना कि खाली MHC और एंटीजन का प्रेजेंटेशन T cells को एक्टिवेट नहीं करेगा. They should have a cell adhesion molecules, right? हाँ. Then what is happening? This activated T cell is doing what? They are inducing the CD4 cells. They are causing the activation of B cells. एक ही चीज से नहीं एक्ट करेंगे. एक्टिवेशन जहां पे भी किसी भी सेल का हो रहा है, they are activating all the types of defense mechanism present there. So they are activating the B cells, which will be forming the antibodies. Antibodies will detect the antigen in the blood itself and they will be destroying. ठीक है? And then inflammation and delayed hypersensitivity is the part of जहां पे भी destruction होगा, inflammation वहां पे होगा ही, right? And also they are activating the cytotoxic T cells as well as, right? So it will depend upon which type of cell is being activated first. It is not that कि अगर cytotoxic T cell activate हुई है तो T helper cell नहीं activate होंगी और B cell नहीं होगी, right? But which one is primarily activated? ठीक है? ये बताओ माइक्रोस्कोप में हम T और B lymphocytes को आइडेंटिफाई कर सकते हैं क्या?